Hi, my name is Love, and I'm an SI for Dr. Maduro's BIO005A class, and this video will cover topic 4. So topic 4 consisted of the 4 major macromolecules: carbohydrates, lipids, proteins, and nucleic acids. So to start off with the major concept, you have single subunit called monomer, and a monomer links with another monomer to form polymers. And this is done through a dehydration reaction where you will release a water molecule. So in the case of this specific polymer right here, we have 4 monomers linked together. In between these 4 monomers, we have 3 bonds. 1, 2, 3. Therefore, 3 dehydration reactions took place, and we released 3 water molecules. The opposite end, in order to break this polymer, it would be a hydrolysis reaction. So we'd have to put in a water molecule in order to break it into a monomer. Moving further, we'll talk about carbohydrates. So carbohydrates are sugars or saccharides, and they provide a quick source of food energy for our body. The monomer of carbohydrates are called monosaccharides and are given by this formula here, where N is the number of carbons and it has to be larger than 3. The basic components of a monomer of a carbohydrate are that it has at least 3 carbons, where one of the carbons is a carbonyl, and the rest of the carbons must have at least 1 hydroxyl group. Then you fill the remaining bonds with hydrogen. So let's go through an example where we draw a 5-carbon monosaccharide. So first step is to find the formula. So in this case, we'll plug in 5 for our number of carbons into this formula. After this, you want to start drawing your monosaccharide. So your first step would be to draw the carbon chain. So I'll draw 5 carbons going down. 1, 2, 3, 4, 5. Second, you want to pick 1 carbon to be your carbonyl. In this case, I'm going to pick carbon 1. I'll put a double bond. And then, as we stated before, the rest of your carbons get a hydroxyl group. So OH on carbon. And since carbon can have 4 things bonded to it at once, the rest of your bonds will be hydrogens. So, and since this carbon only has 2 bonds, you have 2 hydrogens. So this is one way to draw a 5-carbon monosaccharide, where you have your carbonyl at one of the edge carbons. So let's draw another way to draw a 5-carbon monosaccharide. And in this example, I'll draw a carbonyl in one of the carbons inside. So let me pick carbon 2. And I'll fill the rest of my carbons with hydroxyl groups. And finally, I'll throw in my hydrogens. This one doesn't have one because it has 4 bonds already. So we'll move on. So now that we've drawn 2 types of monosaccharides, what is the primary difference? Well, it's the location of the carbonyl. So on our monosaccharide on the left, we have our carbonyl at carbon 1, which is right here. And what's special about that one is that we have a carbonyl attached. So meaning we have a double bonded O attached with the hydrogen as well on that carbon. So this makes this a functional group that we reviewed last video called an aldehyde. And the monosaccharide on the right has another carbonyl on carbon 2. And that carbon has 2 carbons on either side of it, meaning that it's a functional group called a ketone. This is why, if you were to call a monosaccharide with the aldehyde group, you'd call it an aldose. while a monosaccharide with the ketone group would be called a ketone. So now that we just reviewed monomers of carbohydrates, we'll move on to the polymers. So if you take two monosaccharides and link them together, it would be called a disaccharide. And this link will be caused by a dehydration reaction that we talked about earlier and will release a water. And if you take multiple monosaccharides, meaning more than two, and put them together, you will form a polysaccharide, which is shown over here. And it takes the same reaction called a dehydration reaction. So to move on to the polysaccharides, there are two types. You have storage polysaccharides and structural polysaccharides. The storage polysaccharides are mainly used for energy, so they will be broken down by your body or by the plant. And they are made up of polymers of alpha glucose and are stored in plants in terms of starch and are stored in animals as glycogen. On the other hand, structural polysaccharides are used for structure, so they will be relatively more rigid. The polymers are made up of beta glucose, and they're used to build structures of cells and tissues, and are very strong because of its capability to form many different bonds. For example, a structural polysaccharide would be cellulose or cell walls. On the top left is an example of starch, and it is a polymer of alpha glucose, meaning all the hydroxyls are facing down here. And this creates less of a chance to make hydrogen bonds. Therefore, it's easier to break down, which is why we can use it as energy for our body. On the other hand, beta glucose linkages have alternating hydroxyl groups. This allows them to create a lot more hydrogen bonds, which you can see on the bottom right. Having a lot of hydrogen bonds makes it more strong, more rigid, and harder to digest, which is why many structures, cell structures, and walls are made up of beta glucose. So next, we'll move on to the macromolecule lipids. Lipids are large molecules, and they're nonpolar, which make them hydrophobic, and they consist of primarily carbon-to-carbon or carbon-to-hydrogen bonds. The types of lipids we talked about in class were fats, phospholipids, and steroids. So fats consist of fatty acids joined to glycerol by ester linkages. All right, so here is what we have from the last slide. We have a glycerol on the left, and we have a fatty acid on the right. So why do we call it a fatty acid? Well, it's because we have a fat, which is this hydrocarbon right here, attached to a carboxylic acid, which makes it a fatty acid. And the way they're stored in the body is in a form of a glyceride. And the way that happens is a dehydration reaction between these two hydroxyls. And what happens is they dehydrate, and they are linked by what we call esters, which look like that. Having one fatty acid to a glycerol is called a monoglyceride. Having two would be a diglyceride, and having all three filled would be called a triglyceride. So there are two types of fats that we talked about in class. We talked about saturated fats and we talked about unsaturated fats. Saturated fatty acids are fatty acids that contain only single bonds. As you can see in this picture, they can pack a lot more tighter, and therefore at room temperature, they are solid, which is why butter is solid at room temperature. On the other hand, unsaturated fats are fatty acids with at least one double bond. So in this case, they're not able to pack as tightly because this double bond creates a kink. Therefore, a common example is oil, and which is why oil is liquid at room temperature. So the next type of lipid we'll talk about is the phospholipid. And the phospholipid contains a polar head group because it has a phosphate. And the tail is a non-polar hydrophobic tail because it consists of hydrocarbons. Therefore, if you put a phospholipid in oil, the tail will rotate to the oil. And if you put it in water, the head will face the water. In terms of biology, we talked about the plasma membrane, and the inside and the outside of the cell contain a lot of water. Therefore, the heads would face the ends, while the middle is the nonpolar side of the plasma membrane. So next we'll move on to proteins, and the monomer of proteins are referred to as amino acids. The amino in amino acid comes from this amine group on the left, while the acid in amino acid comes from this carboxylic acid on the right, while the carbon in the middle is an asymmetric carbon that's attached to a side chain called R. There are 20 different amino acids, and each amino acid is characterized by its side chain. Amino acids are grouped based on their side chain properties. Some are hydrophobic, some are hydrophilic, some are acidic, while others are basic. Hydrophobic amino acids are characterized by their side chains, which have mostly nonpolar qualities. So that's why you see mostly hydrocarbons in this case. So you see H here, CH3 here. While some may think tryptophan has polar qualities because of its nitrogen over here, however, it's dominated by a hydrocarbon cyclic ring, which makes this amino acid overall hydrophobic. As with polar amino acids, you can see that these amino acids side chains have very polar qualities. So you see things like hydroxyls, which you see in many of them. You see amino groups, and you also see a sulfhydryl group. These are all polar, and most of them could hydrogen bond. What makes them different from acidic and basic side chains are that acidic and basic side chains have the capacity to become charged or ionic. In the case of acidic side chains, you see that there's carboxylic acids, which is why they're called acidic right here and right here. While basic side chains consist of NH bonds, and they're either NH+, NH3+, or NH2+. So amino acids are the monomers to proteins, and they are formed through the same reaction as we talked about with the other macromolecules, called dehydration reactions, where we release a water. In the case of amino acids, they're linked together by a bond called peptide bonds. Also, something notable to note is that proteins always start with the amino group, and each subsequent amino acid is attached to the carboxylic acid side, creating the polymer. So what are proteins? Proteins are polymers of amino acids, and they're the most diverse macromolecules. Proteins are involved in many different processes in our body, such as metabolism, transcription, and even translation. And their folds determine their characteristics, which is why we say that structure determines function. The primary structure is the first step in protein folding, and it is called the single polypeptide chain, which is a simple linkage of all the amino acids present by peptide bonds. The size of the protein is determined by the length of this polypeptide chain. So next, we have the secondary structure of proteins, which consists of beta-pleated sheets and alpha helixes. It's important to see that a single polypeptide chain can have both beta-pleated sheets and alpha helixes. So these are held together by hydrogen bonds between the carboxyl groups right here and the amino groups in the backbone, not the side chains of the amino acids. So next, we have the tertiary structure of a protein, and these are held together by side chain interactions. So we can have hydrogen bonds, we can have hydrophobic interactions, we can have disulfide bridges between the cysteine amino acids, and we can even have ionic bonds between the acidic and basic amino acids. Tertiary structures kind of determine the overall shape of the protein, and the key note to remember is that this is still one single polypeptide chain. It's just folded up because of all the different interactions it's been through. Finally, the quaternary structure is when multiple polypeptides interact together. So in the picture on the left, we have two polypeptides coming together to make a protein, while on the picture on the right, we have hemoglobin, which has 4 different polypeptides coming together to make it one protein. So this is just a little diagram to summarize the major macromolecules and their monomers and polymers. So to start off with, on the left, we got amino acids, which are the monomers of proteins. Moving on, we got fatty acids, which we talked about, and they form ester linkages, and they form the polymer lipids. And carbohydrates, the monomers are monosaccharides, which are sugar residues, and they combine to form polysaccharides or disaccharides, which are carbohydrates. And finally, a little quick review of the next video, we have nucleic acids. So nucleic acids are made up of nitrogen bases, and they combine with a sugar, in this case a ribose, and a phosphate in order to become a nucleotide. These will combine to form nucleic acids and eventually link up to become DNA and RNA. And that is all for this video. Thank you and good luck this week.